And let me get rid of the echo. Oh, we're live. Holy shit. Hi, boys and girls. We're live here with Detzel and Orge and Scott.
And we have an immutable in the side chat there.
Yeah, where's the immutable?
He says, are y'all going live soon? I'm not interested in neutral theory, so y'all can hammer that to death.
Well, you know, immutable, I'm not sure if we're interested in you.
Well,
so what? How did you want to order this? We have too many topics for a couple hour stream, so I'm gonna let you figure it out.
Yeah, so basically what I want to do, it's going to be, for me, it's going to be neutral theory at your request, and also the stuff that I had about abio, which is why Immutable should join, because I specifically was reading some stuff to give him some feedback because he was looking for that. So, you know, he might not be interested in neutral theory, but that's going to be a quick topic. The rest of it can be abio as far as I'm concerned.
Yep.
To CRISPR, I agree. It's a good paper by Jensen. I think that there's still some pause that's required because something I talked over with Dan is the fact that both the Hahn paper and the Jensen paper are using human genome data in order to discuss this. And I pitched it to Dr. Dan. You know, we've been talking about this quack idea of genetic entropy for a long time. And one of the things that's come to the forefront of this discussion is the fact that in the human population, there is relaxed natural selection because of industrialization and healthcare. And if that's the case, then maybe the human genome is not the best place to look in order to gauge the relative effects of these different forces. You know, if we're trying to determine, you know, which one is more of a primary force in shaping evolutionary change, neutralism versus selectionist models, you know? So I pitched that to Dan. He agreed that he thought that was a good point. So, you know, I likewise would say to CRISPR, take that point and do with it what you will, because I think that there's further research that'll have to be done. But I'm definitely not, after reading a bunch of this by now, I'm definitely not like a hardcore anti-neutralist guy. It's just that I still have a sympathy toward the selectionist program. I think that still has something to offer, and I'm still sympathetic to the idea that it's the primary force shaping evolutionary change through time. I may change my view on that, you know, as I continue to study, but that's still where my sympathies lie.
So, Detzel.
Yeah.
Could you real quick run over the competing theories that we're talking about here in a quick little blurb so I and the rest of everyone understands?
Sure. All right. So basically you have the neutral theory, which was really put forward by Motu Kimura. And what that is saying is that at the molecular level, the vast majority of evolutionary change that takes place is neutral. It's not being, it's not subject to natural selection, and rather drift is the main player. And then to contrast that with the selectionist program that says, nah, bruh, selection is still the main game in town. And so that's kind of that's the dispute here. Now, the neutralists are not saying that natural selection doesn't happen or anything like that. What they're trying to say is that at this particular level that we're talking about here, the molecular level, that drift is a main player. And they also, there was a corresponding point I wanted to make here, and I'm losing it in my head. Hold on here. Yeah, I see CRISPR talking here. Neutral theory is not a quick topic because there's since the late 60s all the way up until today, you're going to find a ton of literature. So Kimura first put it forward in the late 60s, and then he had a review in 1991 where he kind of like, you know, put forward his case once more. And there have been a bunch of supportive articles and critical articles. You have Gillespie, you have Hahn and Kern, this particular paper that we've been talking about a lot here. And then the rejoinder to Hahn and Kern is the Jensen and Lynch paper. And they're really going back and forth on a lot of this. I contacted Hahn after I received Dan's response linking me to the Jensen paper to get some of his feedback to see, because he did not publish a response to the Jensen paper. And I asked him why and what his thoughts were. And he responded back saying that he didn't respond because they agree with 99% of what the Jensen Lynch paper had to say. The only disagreement that remains is whether or not we're really going to consider background selection to be a kind of selection. And furthermore, he linked me to a follow-up paper having to do with HIV and the evolution of HIV in terms of like how it relates to this notion of soft selective sweeps. But there's a confusing element to this, which is that in the Jensen paper, they say that the Hahn-Kern approach, the selectionist model that Hahn and Kern are putting forward, they say that this is easily to be considered a minority viewpoint. But when Hahn responded back to me, he said that his supposition was that no, Jensen-Lynch are the minority viewpoint. And Dan responded back to this point saying, I'm not really sure whether or not we can say anything about minority viewpoint. It might be something more having to do with, taking this more as a case-by-case basis on what species we're looking at and stuff. And I really sympathize with that point of view because I gave you a link also to the paper. Let me see if I can find it. This is what I get for having like 40 different papers open. But CRISPR will know what I'm talking about. Neutral theory, the null hypothesis paper. It's a popular, quick article that is referenced frequently on Swamitdas's science forum. And in that article, it likewise says pretty much what Dan says, that the importance of which is going to be the primary factor in play, whether we're talking neutralism and drift or selection, may really start to come down to like looking at it at a case-by-case basis, which is also why I'm suggesting that we might want to pause in using human genome studies to determine this, because part of Jensen and Lynch's response relies on human genome studies. But if it's the case that selection is muted in our population, that might skew your results. And so that would be a partial rejoinder that I would have to what Jensen and Lynch had to say.
So Hahn and let's see, was it Kern? No, let's see. Yeah, Kern and Hahn. Did they use human?
Theory, but that would be a quick topic.
Did they use human genomes for their study? I think somehow. Are you there, Detzel?
Oh, that's all.
We lost Detzel.
Yeah, sorry. One sec. I had some feedback that I wanted to get rid of. I tried jumping into the live stream chat, and our stream started to play out loud, so I was trying to fix that real quick. Okay. So can you repeat whatever question you threw my way?
Okay, Kern and Hahn, did they use the human genome in their study?
They use some data from the human genome, and one of the good criticisms that Jensen, Lynch et al. Have of the Hahn and Kern paper is that part of their evidence relies just on looking at the coding, just at looking at the coding areas of the human genome, not any of the non-coding areas. And that's not to say that all of their evidence relies on that, but some of their data is just looking at the coding segments, and that is just no go. Like if you're really going to try to say selection is ubiquitous throughout the genome, you need to take a total view of the whole genome and not just look at the coding segments. So
I'm trying to get my head around why there's an either or thing going on here at all, because neutral theory pretty much covers the this most of the genome that is not in, basically it's not. I don't want to say it's not important, but it has very little any neutral mutations basically happen in those introns and all those areas that, I mean, that's where a lot of the variation comes up, and some of it comes up in the genes, and that's kind of what Kimura was targeting. And so you have that, and then of course you have the highly conserved regions which aren't going to mutate as fast, and when they do, so basically there's a possibility of some evolution happening there, some change happening there, right? If it's a non-neutral change and it's beneficial. So, but those have got to be much rarer, right, than the non-conserved regions changing. So why is there this either or thing going on between these two ideas?
So yeah, I mean that is a good point. And it's really just, I think it kind of has to go with like the flow and ebb of fashions and paradigms over the course of the 20th century.
Right.
Because basically, like you saw this pan-selectionist program that was in strong operation earlier on, and then you had the pushback against that.
Right.
That was like that was made most famous with like Gould and Lewontin's spandrels paper.
Yeah.
And then this is basically the whole topic that Massimo Pigliucci talks about in this paper that I have posted in the side chat of like the rise and fall of these paradigms. And he talks about how you had the pushback from the anti-adaptationists, but then after the anti-adaptationists had their heyday against the adaptationists. I'm sorry, am I cutting out?
A little bit, yeah.
Okay.
You're good.
Okay, I'm good. So yeah, it's just like this rise and fall thing of these competing programs. But Massimo Pigliucci, in his recent, well, not totally recent, but somewhat recent article that I have posted here in the private chat, is making your point, Speed, which is he's anticipating now that everybody's gotten their little tiffs out. Right. We can finally get down to a more pluralistic approach.
Yeah, an integrated approach between the two ideas.
Yeah, but I still think that it will be, it's still important to consider whether or not there is a main driver, because I don't think anybody disagrees. And when I say that, I'm talking about professionals. I'm not talking about myself, because I'm not a professional. I'm just an onlooker who's studying the issue. So I'm not pretending as if my vote counts here. But when I'm looking at what the professionals are saying, it seems that nobody disagrees that all of these forces are in play. It's just that it seems that there still remains some disagreement over whether or not we should consider selection to be the main driver. And there are some people who want to say that, as a matter of fact, drift is the main driver. I think that Dan has recently said, like, for example, when he debated Standing for Truth recently on Neff's channel, it seemed like, if I remember correctly, he was stressing that he believes that drift is actually the main player.
Yeah. Yeah, and it's like in ecology, there's also a fight going on between neutral theory and niche theory, and it's quite a fight. And I think neutral theory may be winning that one, but I'm not sure. But that has to do with so I don't understand it totally, the ecology, but there's a lot of drift, and they've done some remarkable tests. There's a paper I'm going to find on that, and I'm going to look into it a little more, but that is kind of interesting. The difference between these two paradigms, and neutral theory seems to be winning there. But yeah, I think it's a merger, and I don't, and it always goes over my head when people fight over one theory or the other, which is the main driver. And it could be that one thing will be the main driver in one species, another thing in another species, and then within the species, one particular segment of the genome, you'll find that one is the main driver and the other is not. But neutral theory accounts for a lot of the variation that we can't account for by selection alone. And it answers the question of why is why don't pigs have wings, that sort of thing, right?
Also,
Yeah, I think
there's such a either or kind of attitude that people get in arguing these theories. It's possible that multiple theories can be true, but you have to have things in there that keep you rigorously testing the theory that you have. Even and people tend to want things to be simple, and life isn't always simple. But how do you make sure, if you're going with a kind of a both and attitude, that those theories are rigorously tested?
Yeah. Yeah, well, I guess that's how they get tested. They kick them around and they fight, and everybody's got their territory or their favorites and they fight it out. But I kind of sit here at the top and say, hey, can't we all just get along? I don't know. Anyway, okay. So what was it in Hahn and Kern? What did they come up with that was such convincing evidence? Did you get your mind around what they actually found?
So one of the things that struck me about the Hahn paper was that they did this analysis where you can basically test to see whether or not there's any indication of selection. And one of the evidences that they gave, which to my, if I remember correctly, isn't really contested by Jensen and Lynch in their response paper, which is that when they looked at the genome, what they found was that no matter where you looked, you found indications of selection nearby. So any random loci you would look at, nearby there was indications of selection, which suggested that selection had a major influence on the genome overall. And that was an interesting argument to me. And I looked at the Jensen paper, and while they did effectively rebut Hahn and Kern in terms of the whole thing on them just looking at the coding segments, nevertheless, if I remember correctly, I didn't really see a response to that. They seemed to kind of like take a wary approach to that, of not having a comment on it per se at the time. But I'm just going by memory here. So I might, I'll look to see about that again. But from what I remember, I didn't really get like a firm response from the Jensen Lynch paper about that particular data piece. So
right. This morning I read Kimura's conclusion to his, 1983 book, Neutral Selection Theory. And he was he seemed to be on the fence as I am. That both things were important. Of course, this was his favorite theory, and this was back in 1983 or whatever. But that was interesting. But I think we need to look deeper and probably go on to the next topic here.
Heard. Yep. And the last thing I'll mention here is you were talking about ecology, and today I came across Aviator Nevo, an Israeli evolutionary biologist. And his thing has been focusing on this particular area. I don't know, he calls it like the evolution canyon or something like that, where he's basically like been documenting all sorts of kind of speciation and studying ecology and stuff like that. And he has an article that he wrote for the Encyclopedia of Biodiversity, in which he discusses the dispute between the neutralists and the selectionists. And it seems like it might be like one of the minority voices, since you're talking about ecology and how it seems like neutralism is winning the day there. He seems to be saying that he wants to grant neutralism its place, but it still looks like he wants to say that selection is the main driver. So he might be a voice in the ecological wilderness on that one. I'm not sure, but I did stumble across him today.
Okay, so we'll look further into that. And I do want to watch these. There's some MIT lectures on neutral theory that I want to pick up on, and we can probably revisit this at some point. Okay, so enough of that topic. Moving on.
Yep. So my next thing is on abiogenesis. And so this is really the stuff that I was hoping for more interaction with, because with the neutralism thing, it was kind of at your request, Speed. This is something that I'm still studying a lot, and I have a lot more to learn on this issue. Same thing with abiogenesis. It's just that I had some real feedback on this topic. So the first thing is that you see, I think it's recently been said that Tor has been putting out his videos, and one of the things that he's been talking about is the fact that initially we thought we just had just a reducing atmosphere in the early Earth, and that this basically like precludes any kind of synthesis of organic molecules. I think you mentioned that was part of his criticism, if I remember correctly.
Yeah, that it was more of an it was less of a redox atmosphere than it was. There was more oxygen present at some time early in the Earth's atmosphere, according to some research they did on zircon crystals, basically, and that's a paper that Tour cited. So.
Yeah,
but they also said that this was a periodic thing, and that there were probably times when it was a reducing atmosphere as well, and that was his main pushback. But it didn't carry much weight as far as I could tell.
So the reason why I'm bringing that up, because that's like, it's not just something that that's exclusive to Tor. You could trace that objection way back. Like Jonathan Wells has been saying that for years too. You can find that same argument in his old book, Icons of Evolution. The thing though is that, and Jackson Wheat in his Abiogenesis Part 2 makes essentially the same argument that a lot of abiogenesis researchers are now looking to the oceans because they can be more confident in a reducing environment there than on land. And this is something that a lot of people do. But I'm bringing this up because one of the papers that I sent you talks about basically the successor or the I'm not quite sure what word to use, but there's a researcher who studied under Stanley Miller, and he went on to do a lot more research in this same area, and he repeated a lot of the same experiments that Miller did. And what he decided to do is he decided to see whether or not he could reproduce the same results even with oxygen present. And one of the results that he lists in the article is that, in fact, even in the presence of oxygen, they were able to yield comparatively smaller amounts, but still non-trivial amounts of basically alanine and glycine. So, what was originally taken to be impossible, which is basically synthesis in the presence of oxygen, turns out to be not so impossible. And so this kind of opens up the gateway again to the idea that abiogenesis could occur on land, and you don't just have to look to the oceans anymore, which I thought was helpful to Diemer's viewpoint, because he wants to get away from the oceans. He wants to get back on land. He wants to return to Darwin's warm little pond again. And so if you have the oxygen objection as still a live thing, that would be hurtful toward Diemer. But according to this research and repeats of Stanley Miller experiments, it looks like you can, in fact, get yields even in the presence of oxygen.
Yeah.
From what I've read of Diemer, and I'm not completely through with the paper yet, it doesn't seem like he's completely objecting to marine systems like that could be like habitable for the origin of life. He just seems to want to push it away from like deep sea hydrothermal vents instead of.
Yeah, in Part 2, he allows for formation of some biomolecules in the ocean, and basically washing up, with tides onto the shores, and then ending up in these pools. So there's kind of a mix of both. And I think that is often missed in all of this, that there were many cycles, and even the oxygen redox cycles were probably present, so that you had a little of both at different times. So,
yeah, I think he's proposing an aquatic system that kind of oscillates back and forth between terrestrial and aquatic, just based upon climate cycles, if you want to call it that, or weather cycles that would be present on the early Earth.
Yeah.
Yeah.
Well, he talks about 1 million test tubes or 1 billion test tubes or whatever. So, anyway.
One of the things that he does bring up, though, is the dilution problem, besides the water problem. And so, I mean, with these, though, I want to stress that one of the things that I didn't get done today, it was like one of the last things I was hoping to get finished, but I really started to nod off after reading so much, and I just wasn't able to get it done. But Lane specifically addresses both of those issues in his books. Immutable, I think you have both of Lane's books now, The Vital Question and Power, Sex, Suicide. And
Yeah, I think so.
you know, in terms of, what's that?
I think so. I think you did send me those. Let me check my email again. I think so.
So basically, I would just encourage you to check into that, because that's also something that Demeter wants to bring up. He wants to bring up the water problem. But again, this is something that Lane specifically addresses. Now, I can't vouch for the credibility of his response, because that was what I was supposed to get done today, is go back and review what he had to say on those issues, and I wasn't able to get it done. So I can't vouch that Lane successfully rebuts these issues, but I do know that he specifically addresses them. So it would just be worth looking into what Lane has to say about that. Another thing that I wanted to bring up is that when Demeter is putting forward his theory in its point of no return paper, the way that he argues, it's pretty much an argument from irreducible complexity. And I sent Speed a final email quoting the relevant passage of basically like his argument having to do with information and structure / function, and how they are interdependent, and because they're interdependent, there's no way that they could have independently formed. It's an argument from irreducible complexity, and for me,
I don't know if it's irreducible complexity in the sense that you could have the systems being separated, but you can't have the same type of dynamic that, in terms of like the information that a system carries, without those two operating in conjunction with each other.
Here's a paragraph right here that you quoted. And
yeah. All right, I'll read it real quick. How is it possible to bootstrap the components in Figure 1 and set Darwinian evolution in motion in natural conditions? All three components, the information, the structure, function, and the rule between them, are mutually dependent for their existence. That is, the components are attributes of each other and cannot be analyzed independently. If we take the information, for example, it cannot exist without resolving the structure function component. Otherwise, the string of units, nucleotides of RNA, would be random without information. It would be like the working of a mechanical clock without installed hands. You would never know what time it is. I mean, this sounds very much like Behe. And my problem here is that if you look at some of the metabolism firsters, like take a look at Freeman Dyson, one of the ways that he'll talk about this is that, his view is that you have a metabolism first situation, but RNA develops over time as well. And because lipids are permeable to RNA, you basically get this endosymbiosis where the RNA infiltrates the membrane, and then from there uses the organic material to perpetuate itself through time. And that would be, you have independent lines of development that have nothing to do with one another until this merger takes place.
And that's what we were arguing about today, Immutable.
I don't see that as consistent, though, because I don't, for instance, with that kind of analogy, right, I don't see why in a protocell it would have the molecular machinery needed to, or that can perform something like replicating RNA if RNA was never part of that system to begin with.
But the only thing that's necessary in a protocell, a pre-protocell, is for it to grow, for the lipid membranes to grow, and for it to divide with its cargo, basically. So diffusion and dividing, and that's all you need. You don't need information at that level, I don't think.
No, but the thing is, okay, my contention is I think you do. Like, you could argue, for instance, that there's some kind of like stochastic element that, okay, there's no information, and the system is kind of just feeding upon its environment in terms of how it's replicating. But that doesn't seem like it's going to yield a faithful replicator to you.
No, it shouldn't. In
Right?
fact, that's the beauty of it.
No, but
Because
I mean that's
because
for me,
then at the point of the merger, of the two systems of Demeter's system merging with these micelles or with these protocells, then you would have something interesting.
Yeah.
Your information would merge, and then we would have a different kind of replication, right?
And
But
one of the things that one of the things that I wanted to point out here, because a part of Demeter's presentation that I thought is brilliant, and the article that he references to this point, where he talks about basically an RNA peptide world, and how when you have these two things together, when they interact, they form more stable structures. So it's only at the point of that it's at the point of the merger that you can get this stability. So I mean, I would point out that, if that's the case, if you can actually get this stability when they start to interact with each other, that they don't have as much in their absence. With respect to the metabolism first thing, you would be looking more toward the environment to produce the kind of metabolic activities. And then since, with Lane, he's going the hydrothermal vent route. So you have the protocells forming in the pores of these vents, and then the interaction between the base ocean, the base water versus the more acidic water, driving these chemical reactions. So I think that is, I don't think that Demeter's argument that this all necessarily has to develop together and in one go, I'm not quite sure I want to buy into that. But what I do really like about it is his point that you get this kind of stability when they interact with one another.
To me, I mean, I think you can explain metabolic models in terms of that hypothesis and framework. Because if you're dealing with a system that's dealing with a protocell and you have peptides of some length being synthesized, some of those peptides may possess properties that allow them to be embedded into the micelle once it divides. Some of them may have some degree of catalytic activity in terms of having an affinity towards catalyzing some reaction. And the more that happens, right, the more energy a cell is able to utilize, the more complex those structures can become. And which structures get selected for is directly determined by its environment and the types of peptides that happen to get produced.
Yeah, I don't know if Demeter is actually making that strong a claim about, though he does push back a little on metabolism first. Like I told Demeter today, we were on the phone, and I said, just drop the word first and think of altogether.
Yeah, I mean, I don't think Demeter's making that strong of a claim either. I mean, I think it can naturally flow from the type of thing that he's proposing.
Well, I'll be happy if he's not really going down the irreducible complexity route. It's just that the language that he's using here, it just sounds strongly suggestive of it. He does make the complaint in his article that there's no mechanism toward the development of basically heredity for the metabolism first point of view, but he references again Lane and a couple other researchers. An article that they wrote called "The Origin of Heredity in Protocells," and they provide a computer simulation based on the environment that they have in mind and the chemical reactions that are relevant to see whether or not you get to a situation of exactly the type that Demeter is talking about, where you have the emergence of the information structure function relation. And the onset of replication, and their results in that computer simulation were positive. And I mean, like, he references the paper, but he doesn't address the results that they put forward in that paper. So, I mean, that would just be something to look further into too. That paper is called "The Origin of Heredity in Protocells." Once again.
Okay, forgive me.
Yeah, I haven't read that one, so.
Sorry. Anyway, Otangelo claims that we must have embedded proteins in order to have permeability of those membranes. And
No, that's not correct.
you
He
said
also claims, Speed, that gravity doesn't affect gases.
yeah, I know he said that.
Yeah,
Wrong
the
with
Tanner
that.
wasn't exactly the sharpest tool in the shed. I hate to shatter that illusion.
Famously.
You
But
still-
but you mentioned that those lipid membranes are permeable to RNA,
Yeah.
and so and also to some small peptides as well, depending on which peptides they are. So yeah, that's not the whole story. And then there's there's also the possibility of ingestion, of membrane ingestion or whatever. Anyway, I just thought I thought that might be something that we needed to address, but other than that, we're not going to listen to Otangelo much more here. So, but he always comes up with a topic. No evidence that protomembranes should meet the requirements or whatever. So, carry on.
Yeah, this paper I can't read too much of because a lot of it is like really mathematically heavy.
Which one?
The origin of heredity in protocells.
Who's the first author?
I think it's Lane. No, Timothy West, Victor Soho, Andrew Pomianowski, and Nick Lane.
Okay, the origin of heredity in protocells and the philosophical something or another. Royal Society?
Yeah,
Yeah, let me check.
Nick Lane. Okay, good. Okay.
Yeah, Royal Society.
Yeah.
Yeah, a lot of this is like really mathematically heavy, so I'm not sure.
Let me take a look.
Yeah, you get. I'm sure it's 13 pages though, so I mean, it is math heavy. I looked for their results from their computer simulations to see whether or not it looked like a happy ending. I haven't gone through the detailed math, but since it's 13 pages, I think that I
can give them the benefit of the doubt on it. It's let's see. The results show that's under impairment.
If you just, I think that if you just take a deep breath, you'll be able to do that. 13 pages, even if it's heavy math. And I'm scrolling down right now, and actually I'm not seeing a ton. I see a lot of graphs and stuff.
The math didn't look that bad.
The math gets into it like near the end when it starts getting into like the methods they use.
But yeah, in these, let me see if I could scroll down to results.
Yeah, I think it's page. No, it's not. It's page.
So just to quote it really quick, the results show that under certain parameter ranges, positive feedbacks can indeed drive protocell growth with tight amino acid binding. The mean crystal size falls relative to protocells with weak amino acid binding. Accordingly, the concentration of crystals in the cytosol increases in protocells with tight amino acid binding. This is not what I was looking for, though, because it specifically addresses the whole production of the emergence of heredity. And they say that this indeed happens as well.
What page is results on?
That
It's
was,
on page 4.
I started results. Yeah, I started results on page 4. But let me just see here.
Oh, there's that. Okay. Oh, wow. What did I share? What the hell did I share? I hate myself sometimes. I shared my Mac screen with all the little pictures on it. And I had your address on there, your home address and your telephone number immutable.
Well, thank you. You keep doxing me on the weather report, so.
No, I didn't really have that on there. I did have a picture of you in the tub when you were a child, though, that your mom sent me.
I appreciate it. You'll be getting a letter from my lawyer soon.
You're taking it up with your mom. Okay, so here we go. So now we actually. What? No, that's not it. That's West and Lane. I cannot get my shit together here.
Oh, that's.
Some of these catalytic rates they're reporting seem to be low, but I could be off with the literature on that.
The origin of heredity. in protocells. Yeah,
Well, actually, yeah, it's not that bad.
it is. I did have the right one, didn't I? Did I have
Yes,
the
you
right
did.
one? I did. But for some reason, oh, that was a tab.
I just, I'm going to satisfy myself with the abstract right now instead of going through the whole thing. But yeah,
And I don't have my, for some reason, this is not working. I don't know why. I don't know why.
well, I'm just going to satisfy myself. I'll just read the abstract if it's okay, really quick. And
Yeah, go ahead.
So, I mean, here we develop a computational model that examines one of the first major biological innovations, the origin of heredity in simple protocells. The model assumes that the earliest protocells were autotrophic, producing organic matter from CO2 and H2. Carbon fixation was facilitated by geologically sustained proton gradients across fatty acid membranes via iron-sulfur nanocrystals lodged within the membranes. Thermodynamic models suggest that organics formed this way should include amino acids and fatty acids. We assume that fatty acids partition to the membrane. Some hydrophobic amino acids chelate iron sulfide nanocrystals, producing 3 positive feedbacks: an increase in catalytic surface area, partitioning of iron sulfide nanocrystals to the membrane, a proton motive active site for carbon fixing that mimics the enzyme. Was that ECH? I have no idea what the hell that is, to be honest.
Yeah, I know.
These positive feedbacks enable the fastest growing protocells to dominate the early ecosystem through a simple form of heredity. We propose that as new organics are produced inside the protocells, the localized high energy environment is more likely to form ribonucleotides, linking RNA replication to its ability to drive protocell growth from the beginning. Our novel conceptualization sets out conditions under which protocell heredity and competition could arise and points to where crucial experimental work is required.
And that would be a nice little mini laboratory for Demeter's stuff to work in, wouldn't
I'm
it?
curious what the I'm curious how they're modeling this though, because they mentioned crystallization. And one of the things, like if you've done any bit of like spectroscopy at all, is that usually crystals tend to form under like extremely low temperature conditions, and that they are like very sensitive to the presence of ions and the presence of like temperature fluctuations. So I'm not sure
How are you
exactly.
defining low temperature in that?
Uh,
Are we talking about different crystals that I'm thinking about?
well, if you're talking about like crystals of like amino acids and stuff like that, you need it to be. I can look it up. But no, you're not talking about like inorganic crystals, which form at like asininely high temperatures.
Okay, gotcha. Yeah,
Yeah,
never mind then.
Crystallization temperature of amino acids. I think it's pretty low. I think it's actually around. I know it's below room temperature. At least I'm pretty sure it is. And so I'm curious, like how easy it would be to get crystallization.
I just want to. I'm asking creation myths and CRISPR what the hell this enzyme ECH is.
It's enoyl CoA hydratase. And let's see,
Okay.
is an enzyme that hydrates double bonds between the second and third carbons on 2 trans cis enoyl CoA. So what is that?
Yeah, it's been a while since I did.
CoA. And I don't know why was that important, because let's see, ECH of rats is hexameric protein, which leads to the efficiency of this enzyme. The enzyme, okay, what is it? What the hell does it do? Fatty acid metabolism. So something with fatty acid metabolism is how human bodies turn fats into energy. I don't know why that would be important. Why is this important?
It's just it was mentioned in that abstract there, and I didn't want to walk away from the abstract without at least having an idea what the hell they were talking about there.
Yeah. Anyway, that's what it is. So.
All right, thank you. I appreciate that. So yeah, I mean, you know, I want to at least close this whole thing out, at least my portion of this, by saying that I really like Demeter's model. I'm just not convinced by his arguments so far, to the effect that, you know, he's trying to rule out a bunch of these other models, such as certain metabolism first, you know, approaches. And so far, the kind of arguments that he gives, it sounds to me like he's going an irreducible complexity route in certain ways. He also mentions problems of polymerization, which, Jackson has an article on his abiogenesis part 2 about polymerization increasing by 50-fold by a thermal gradient. So I don't think that, I think that polymerization by these alkaline vents works just fine. He mentions the water problem, the dilution problem. These are things that Lane directly addresses, but I have to at least hold my verdict on that because I can't remember. I remember that I remember Lane addresses them. I just don't remember how he addresses them. So I can't attest to the plausibility of his response. And
Okay,
that's
I
where
think
I'm at right now.
if Demeter could figure out where you lived, he'd probably strangle you for saying that he was associated with irreducible complexity. So we're gonna make sure he doesn't find you. But I think his point was that his model was more plausible in terms of what he's talking about. What he's talking about is the RNA peptides, basically part of this, right? And it seems to me, I don't know if you guys took my point, but what they're talking about here sounds like a wonderful laboratory for Demeter's RNA peptides.
Yeah,
I think Demeter's paper actually serves as a good foundation for one of the areas that his model could be tested at as well. I mean, Deemer seems to suggest that continuous wet-dry cycles would lead to high polymerization of amino acids.
So I'll definitely start working on the restraining order after this stream is done.
Yes, you probably should. Because
but if I'm misunderstanding him, then I hope that he'll just speak up at some point so he can correct me if I'm wrong. That it's just the language he's using and the kind of argumentation here just sounds really reminiscent of Behe. So. You don't want him sending his. But the last thing I'll say is that I still do want to say that I do like his model, particularly because of this work that he's putting here about RNA and peptides and their increased stability together like that. I think that's phenomenal because up until now I had been working kind of on this Freeman-Dyson assumption that they had to have independent lines of development. But what he has effectively shown with his model and with the article that he referenced, that's not the case, that it doesn't have to be independent at all. It's just that I don't think that he's effectively ruled out the possibility that they could be independent. So that's where we would part ways. But I at least very much like the fact that his argument produces a perfectly good rationale for why they don't have to be independent, and I'm happy to go with that.
Well, he has dispatched his attack parrot. He just emailed me, so his parrot is pissed.
Is your understanding the same as my understanding regarding Metabolism First, in the sense that a lot of what I see from Metabolism First seems to be that life centers around, kind of captures the process of autocatalytic cycles, regarding the origin of life, in order to kind of get its kickstart?
Yeah, with proton gradients producing protein motive force, and this is basically there are several Metabolism First models, and a couple of them that I'm not familiar with have been gone over by John Perry in his short little Stated Clearly video on what is
Metabolism. I saw the video he did several months ago on Metabolism First, and he referenced an interesting paper. I think it was on a primitive version of the citric acid cycle or something like that.
Yeah, so they got a citric acid cycle, and there's several other really ancient metabolic pathways that they focus on. So each one is a candidate that different Metabolism First researchers will argue over. But the main two that I'm indebted to and that I rely on are Freeman-Dyson and Nick Lane. That's basically the majority of stuff that I know about abiogenesis, and particularly the Metabolism First model, comes from those two guys.
I would. I'm actually getting a little bit more interested in Sutherland, because I think Sutherland is taking an approach that's actually more agnostic to abiogenesis, and that he doesn't seem to really affiliate himself with any of the current models, but he is interested in trying to see what organic compounds can be can be derived like from certain feedstocks, like certain like cyanide molecules or other molecules that are known to be in very high concentration on the early Earth.
I'll definitely look into him. I'm not as familiar with him. And I want to throw out there, just to try to protect myself a little bit from Demeter's parrot,
Right.
that I wanted to say that I was listening to his presentation that he gave with you and Aaron Raw earlier today, Speed. So I listened to his presentation where he gave his theory. And I'm just like sitting there as he's putting forward his whole thing about RNA and peptide stability. And I'm just like, my jaw's pretty much on the floor, because that was just like kind of like a revelation thing to me. I'd never thought about it that way before. And like, I'm just like noticing that, well, I mean, just Aaron just looked totally like, I have no idea what the hell's going on kind of thing. And I thought it was like a real big shame, because he, like, this moment was like really awesome, that he was putting forward this idea that I thought was highly provocative and original, that I had never thought of before. And nobody around him was actually able to like discern what the hell just happened.
Right.
So, yeah, but anyway, so yeah, try to throw out that compliment, so that way the parrot won't eat me.
Won't attack you. Okay. I'll
So,
try to call out the parrots.
for you, Dessel. A lot of some of the criticism that regards metabolism first hypotheses is that so some of the conventional approaches that that biochemistry uses is, for instance, is using ATP, the hydrolysis of ATP, in order to cause certain reactions to go forward that otherwise wouldn't exactly want to go forward. It's this kind of, it's a way for the cell to compensate for an otherwise unfavorable reaction. But one of the critiques regarding like certain metabolism studies is that you may not have had the ability to perform that kind of reaction, like hydrolysis of ATP, and you may have to resort to something more primitive, like a thioester to amide bond kind of formation or something like that. And so, kind of the fact that the earliest life we have, for instance, it in some sense it seems to be kind of masking its metabolic fossils, if there's any that haven't been, untouched or unscarred, currently.
Yeah. I think the only metabolic fossils that are extant is the use of metal ions, and
I mean, I think that's also a new kind of thing too. If you think about it in terms of like the last universal common ancestor, because a lot of enzymes they use metals, but they don't use metals in their raw form necessarily. You see things like porphyrin rings and various specialized organic compounds that coordinate very specifically with certain metal ions in order to kind of relax and constrain their reactivity.
Yeah, one of the things I'm interested in is very small, the smallest possible folded peptides that can incorporate metal ions as an active center. I don't know if there's any research on that.
I think you would have it more be kind of like primitive ribozymes, if you will.
Yeah.
Because like especially RNA strings that are single-stranded and rather long, if memory's not mistaken, they tend to kind of fold into these very convoluted and beautiful little patterns. And occasionally, when you get this, I call it like clover shape, you sometimes incorporate metal ions into that structure as a means of stabilizing it.
Yeah, Nestleg linked a book which I purchased, which I could go find if I could rummage through a few things. But anyway, I made a pretty big thing of these the interface of geology and early life, and how it is it's a continuum from geology, that it's a necessity of the geology of early Earth that life developed. And
that reminds me, actually, if you're still here, Russell, and you're not wanting to kill yourself by now, Stephen Benner actually takes kind of a pre-prebiotic chemistry approach. He studies what a lot of people kind of consider the abiotic chemistry, so the chemistry like right off of earlier of coming out of the oven. So a lot of stuff concerning if it was possible for precursors to DNA to form in like in the mantle or in certain geochemical settings and stuff like that. And why would you get fixation of phosphate as the molecule of choice to attach itself, in this intricate structure with DNA, as opposed to silicate derivatives or borate derivatives and stuff like that, which happen to be highly present in the in the earth.
I gotta quit shuffling my books. I need a book on shuffler. I couldn't find it, but anyway, go ahead.
There's a book about that on Amazon.
There's a book unshuffling your books.
Yeah, you might want to go in there and order it.
Okay, I just ordered it.
I wanted to ask Immutable, could you send me some stuff from the last fella that you just referenced, as well as Sutherland? Do you have anything by them that you could send me via email?
Yeah, there's some stuff I'm reading from them currently that's kind of interesting. Every now and then, I think every year actually, Sutherland will send out a review paper just kind of highlighting the current findings of his research and the direction he wants to take it with.
All right, cool. And completely irrelevantly, I just wanted to throw in, because I think we're going to end this stream soon if everybody finds that agreeable, because all my stuff is pretty much done. But I wanted to tell Orgate, I've been watching your YouTube channel a lot recently, and I tell you, man, I can't stop laughing. Your material is absolutely hilarious. If anybody wants to laugh.
Yeah, I watched one this morning. Was it this morning? It was good. Yeah, it's funny. Anyway.
Actually, the thing Otangelo is addressing is actually the work of is one of the works of Jack Shostak. He's done work with alternative genetic systems in terms of their replication rates. If they, if you assume that those kind of variations of the sugar could form, and if they could replicate in a manner that's similar to RNA. So you have other sugars like arabinose and threose and other sugars whose names elude me right now. But they tested certain variations of ribose that could be present, and they found that in fact ribose wins out.
If you go look, there's I think this paper we're doing, Deemer or Dahmer, has a couple of links to solutions for the homochirality problem too. So
yeah, part of it is that if you get into stuff like with the astronomy and stuff like that, it's actually quite surprising to find that a lot of comets, I think, and some meteorites actually bear like a strikingly high excess of the proper configuration of amino acids, being the left-handed amino acids.
Yeah. Yep, on the Murchison meteorite, it was upwards of 90%.
Yeah, that's quite surprising. But yeah, that's one of the sources of
90% L for amino acids. Yeah. Wow.
Yeah.
So the creationists are the only ones that find the homochirality problem, as far as I can tell.
It's also important to mention, like with the Murchison meteorites, you also do have other things that are there. You have some purines that are present, although in far less extent. But you do have, I believe, some short chain lipids and some variations of carboxylic acids and other interesting organic compounds.
And they found pathways for the pyrimidine. So
there we go.
I want to throw out here real quick, because the whole philosophy of science thing, just one point on that, which is that I screwed up. I said that Hempel was when I did this video to Dapper Dino, I said that Hempel was an earlier proponent of the hypothetical deductive method, and that's wrong. He was not. He was an early critic of it. My point there was just to stress that hypothetical deductivism precedes Popper. He's not the guy who came up with it. He's just a guy who like popularized his particular model, but it precedes him. And I made the stupid comment that you could trace it at least to Hempel, but Hempel's actually a critic. He's not a proponent. So just for anybody who watched that video, that comment, that was wrong. And that's it.
Okay. We'll dispatch Parrot.
Actually, the conversation Tangelo and Andrew's having kind of leads a few people that are taking a microbial approach to the origin of life suggest, and I do agree with this one, that to understand the origin of life better, we should understand how archaea, the diversity of archaea and some of their composition. Because if life did form in these extreme environments, right, much in the way that certain archaea live very close to these hydrothermal vents and all sorts of insane conditions for life to prosper in, it might give you some clues in terms of what things life could have used in order to get its start.
What I was saying earlier about there being a last universal common population rather than a LUCA has to do with archaea and bacteria at that level. There was a lot of there wasn't a germline heredity way that you could draw the tree. It was more of a bush with a lot of horizontal transfer, merging, ingesting, spitting out, that sort of thing. And that's something that we always have to consider. That if you look at I forget where it was, molecular cell biology or some genetics book I had, they drew the tree of life, and then they drew the part with the archaea and the bacteria and early life as a gigantic messy bush, with everything interconnecting. And
there's a few common depictions that with the tree of life, what you see is that towards the upper parts of the tree, is that it's very well organized. It's this noticeable bifurcating kind of pattern that you observe, but towards the root of the tree, like you were mentioning, right, instead of this nice organized tree with distinct roots coming out of it, near the very bottom of it, where you have prokaryotes and bacteria, or prokaryotes and archaea rather, you have instead this kind of weird convoluted tangled web of horizontal gene transfer that makes it very hard to understand. And detangle that web.
If there was a pathway for one of those cells to basically merge and create something that had a great advantage, then that pathway very likely happened multiple times. So it possibly happened in a population or happened more than once in that tangle and came out. So that denies that there was a one cell LUCA, which always sort of stuck in my, I always had a problem getting over that one, that, there's this one cell that ended up being all of eukarya, but it could have been different. I mean, there are other ways for it to happen. But of course, the creationists fucking love when I say that for some reason.
I'm not sure, but Bessel, this might be an interesting question for you. Do you think, and I'd imagine with metabolism first, the answer would be heterotrophic, but do you think that that LUCA was more than likely heterotrophic or autotrophic?
So Lane actually goes for an autotrophic origin, and there's a bunch of metabolism first stuff that I've seen that goes for the autotrophic. I'm happy with either one. Doesn't matter to me. It does. But the models that I'm relying on, I do have to say that they lean more toward the autotrophic. And I know that there's a big discussion about, how it's impossible that, it could have been an autotrophic first. And I looked into that a while ago, and it seemed like it's really up in the air. Same thing with thermophiles, because a lot of metabolism first research is kind of into the whole thermophiles thing, since we're going by these hydrothermal vents. And some of the reconstructions that have been done of LUCA suggest that it was not thermophilic. And so they suggest that maybe thermophiles arose later after the meteor bombardment and everything like that. But the rejoinder to that was simply, well, I mean, Luca doesn't have to really resemble what the first organism looked like. So,
Yeah, I mean, it seems that you keep in mind that since Luca is the common ancestor for both prokaryotes and Archaea, and Archaea thrive in extreme environments, but prokaryotes do not. It seems that Luca more than likely had to kind of occupy a Goldilocks zone, if you will, of chemical environments. It seemed to to need to tolerate, or the environments that fostered it would have had to be kind of neutral in the sense of like pH or things like that. But I don't know. That's my opinion. It's completely without speculation. It's there.
Yeah, it's something I have to look more into too. I still have, after all of this, I still feel like I've barely begun. But I want an irrelevant question. Wasn't David Neff having this party thing or whatever?
I
was just
yeah.
I think he's going to start it after we're done here, because I think he doesn't want to interfere with our thing.
Okay, we can. Yeah, we can wrap up. I just want to leave with Otangelo finding a huge problem with glucose or something. They always talk about huge problem for abiogenesis.
Oh God, he mentioned this before, and I mean,
huge.
The Mount Everest of problems.
Yeah, and everyone is the Mount Everest of problems. Go out and Google it, for Christ's sake.
If he's going down the glycolysis thing again, if he's going down the glycolysis crap again, this has already been explained to him. Glycolysis seemed to have developed later on, whereas gluconeogenesis seemed to develop first, and there's a lot of reasons for that.
So this was dense, so we should end it. So end the live stream.
To hell with it.
Anybody else have anything they want to say? Anything you want to say to the parrot, or apology?
Goodbye, cool world.
I apologize to the parrot for anything that any of us might have said to offend him.
To offend.
I think all anybody wants to do is offend the
Please
parrot.
don't hurt. Please don't hurt.
Are you assuming the parrot's gender now, Scott?
This channel does not condone pissing off the parrot. Okay. So, we're gonna end it. Goodbye, cruel world.
Goodbye.